Good day, everyone. How are you doing today? I hope you're doing well. Welcome to another tutorial video by the National Fellowship of Christian Pharmacy Students, University of Nigeria, Nsukka chapter, that's UNN chapter. So today we are going to be talking about practical pharmacology, book 3. That is the practical pharmacology that is being done in second semester in the third year. So we're going to be laying some emphasis that will help you to be adequately prepared for your examination and be able to ace in your examinations. So basically, there are a few things you have to understand about practical pharmacology. Now, the examinations could be alternative to practicals. Most of the times, it's usually alternative to practicals, or then you could have life experiments. But usually, so far so good, we have alternative to practicals. So, but any which way it goes, it's very important you get appropriate information from your coordinator so that you can be appropriately guided. Now, one of the things I will have to inform you is that you must read your manual very well. I mean, every single information contained in your manual must be consumed appropriately so that you can be properly prepared for your examination. For example, the conditions that affect the kind of result you get in your experiment must be studied. You must also study the different kind of solutions that are used for different isolated tissues, because basically the experiment you're going to be conducting in this particular course involves isolated tissues. So you need to know the kind of solutions you must use for the isolated tissues. Now, usually what we use here is isolated intestinal tissues. That's for the guinea pig ileum. So the implication is that the solution we use is the thyroid solution. So, but then in your alternative to practical, you may be asked a question that has to do with the uterus, and then you're asked what kind of physiological salt solution, that's PSS, is used in that case. Thyroid solution obviously is going to be wrong there. The answer to this particular question is in your manual. So go ahead and look it up and master that is necessary for other kind of tissues. So the first experiment 1, is what we're going to be looking at, and that is actions of drugs on the isolated guinea pig ileum. Now, it is very important you must have mastered your agonist and your antagonist. It is very important at this particular point you must have mastered your agonist and your antagonist, especially as it has to do with autonomic drugs. That's autonomic pharmacology. You must have been able to understand the drugs that work on the sympathetic system, the drugs that work on the parasympathetic system, the drugs that work on the muscarinic receptors, the nicotinic receptors, and so on and so forth. Because with your basis of the understanding of these drugs, that is how you are able to carry out the experiment, or if it's an alternative to practical, that's how you're able to understand the experiment you're carrying and be able to give the right kind of report you ought to give. So in this particular video, I'm going to be running through experiment 1, the theory behind experiment 1, and I'll be running through the theory behind experiment 2 of this particular course, so you can have a good hold on it and understand exactly why what was done in this particular experiment. You know your normal form of reporting: your title, your aim, your theory, your procedure, your results, then your discussion, conclusion, and then your precautions at the end of the day. So it's very important to pay attention to the instructions from your coordinator so I can able to do it in the right way. So let us look at experiment 1 of this particular course: action of drugs on the isolated guinea pig ileum. Since we are working with the ileum of a guinea pig, the solution we are using, the physiological salt solution, that is PSS, we are using is thyroid solution. You may want to look up the components of the thyroid solution and also have to memorize that, because you may be asked in your examination. So the first part of that action of drugs on isolated guinea pig ileum, we are going to be looking at parasympathomimetic drugs. We are going to be looking at parasympathomimetic drugs. So parasympathomimetic drugs actually talk about drugs that mimic the action of the parasympathetic system, or provokes or influence the action of parasympathetic system. So they make the parasympathetic system to actually act. And then when we talk about the opposite of this, parasympathomimetic, also known as, and the opposite of it is actually parasympatholytic, because it disrupts or stops the action of the parasympathetic system. So you have to go back to your autonomic pharmacology and understand the difference, autonomic physiology, sorry, for understand the difference between the sympathetic nervous system and then the parasympathetic nervous system. It's going to help you to understand the progress you have made so far. So if you're watching this video, obviously you should already be in third year and you should already know what we're talking about when we say parasympathomimetic drugs. So the aim of this particular experiment is to determine the mechanism of action of drugs, agonists of the parasympathetic system on the smooth muscle of the intestine. So there are different kind of solutions that will be provided to you in the experiment. So you could have acetylcholine, you could have acetylcholine, then you could have methacholine, you could have carbachol, you could have neostigmine or physostigmine. Then you'll be provided with normal saline. You'll be provided with the guinea pig ileum thyroid solution. Let me just list them out and then tell you exactly what you are working with in this particular experiment. Test tubes, incubator, and so on and so forth. So, but our focus in this particular discourse here is with regards to the drugs. So we have acetylcholine. Acetylcholine happens to act on cholinergic receptors, where we talk about the muscarinic receptors and the nicotinic receptors. Then methacholine happens to be a semi-synthetic form of choline esters. Are we together now? So the difference between methacholine and acetylcholine is that in methacholine, you already have a methyl group attached to that particular compound. You may want to look at your textbook for you to see that specific attachment of the methyl group. And the addition of that methyl group makes it much more difficult for methacholine to be hydrolyzed by cholinesterase enzymes. Meanwhile, acetylcholine is quickly or rapidly hydrolyzed by cholinesterase enzymes. But then methacholine is less rapidly metabolized by the cholinesterase enzyme. So it's very important to take note of that due to the addition of the methyl group. Then for the carbachol, carbachol has the amide group and it's a synthetic cholinester. It has amide group and then methacholine has methyl group. So the amide group in carbachol, adds a much more good stability to carbachol, making it impossible for cholinesterase enzymes to degrade carbachol. So in terms of the one that in terms of rate of degradation, then you find out that acetylcholine rapidly is degraded or hydrolyzed by cholinesterase enzymes, followed by methacholine, and carbachol is not even affected by cholinesterase enzymes. So it's very important to take note of that. Now, when we talk about neostigmine and the physostigmine, neostigmine or physostigmine simply refers to drugs that act on the cholinesterase enzyme. They are actually anticholinesterase. Neostigmine and physostigmine are anticholinesterase. Simply means that they work on cholinesterase enzyme. They bind them and prevent them from acting on acetylcholine or methacholine. Another under neostigmine and physostigmine, you also have an example like rivastigmine. And we have so many other examples which you can find online in your textbooks and other particular sources. So the neostigmine or physostigmine, they are anticholinesterase drugs. What they simply do is to work against cholinesterase that are captured in the blood or any region where they are found, and then allow for the acetylcholine or methylcholine to act much more longer. Why I'm not mentioning carbachol is because I already told you that carbachol is not even affected by cholinesterase enzymes in the first place. So the addition of any form of anticholinesterase agent like neostigmine, physostigmine, rivastigmine, and the like, pyridostigmine, is not going to be necessary because it's not affected. Now, normal saline just simply refers to such solution of 0.9% concentration. That's weight per volume, 0.9% concentration weight per volume. And that particular concentration is actually the concentration of salt that has equivalent tonicity, that is isotonic with the body or the blood, the body solutions or the body fluid. That is, we're talking about the blood in this particular context. So normal saline, 0.9% weight volume. 0.9% weight volume is the concentration of salt that is isotonic with the body fluid, and in this context we're talking about the blood. So then the thyroid solution, like I already mentioned, the thyroid solution, okay, let me write this. This is anticholinesterase agents or drugs. Then, or you can call it cholinesterase. So thyroid solution in this context is our physiological salt solution, and in short we call that PSS. Like I mentioned, the physiological salt solution you are using is dependent on the tissue you are working with. We are working with ileum, which is an intestinal tissue. The implication is that we are using thyroid solution, and from the start of this particular video I made reference to the uterus, and I made you know that the answer to that particular question, you find it in your manual. So go ahead and study about other kind of tissues and the appropriate solutions that are used for such tissues. Now the test tubes is going to be used for the experiment, and then we have incubator. So let's walk through the procedure of the experiment so I understand what the experiment is about. Then we'll do a few calculations that will help you understand the procedures that were outlined in the manual, and then help you to have a full grasp of how to report your experiment, whether in the real life experiment or in the alternative to practical. So let us make progress to the procedure. The procedure talks about setting up the guinea pig as usual. In your second semester, you must have already encountered how to carry out this process in your first semester pharmacology. So make appropriate dilutions to enable the administration of the various specified doses of the drugs. Now the doses recommended may be increased or decreased to obtain a satisfactory response. So we have doses of drugs that are administered, and it is as prescribed in your procedure. Now, when we talk about dose response relationship in your pharmacology in the first semester, you already understand that when we are working with dose response relationship, we usually work with geometric increases in the dose. So we can start with 0.5, go ahead to 1 microgram, go ahead to 2 microgram, go ahead to 4 microgram, go ahead to 8 microgram, 16, 32, 64, 128, and so on and so forth. So you may want to go back and see how that runs in the dose response relationship. So, but in this particular context, I'm going to be working with what we are asked to do. But any concentration or any amount you're actually supposed to administer is in view of the concentration that is provided to you in the lab. So you must pay attention to that, and that's why we will need to run some calculations so you understand how to carry out those dilutions and do what is necessary. Now, yeah, so let us let's just clean this, and then let's talk about some of the concentrations that you could be given in the lab. So we are working with some concentrations. We have acetylcholine. In the laboratory, you can be given any concentration that the technicians deem fit for you. And in your alternative to practical, they can give you any concentration. So, but what you need to know is to know how to work around the different concentrations and be able to get exactly what you need or desire from your experiment and get the right results. So usually in the laboratory, what we usually have for acetylcholine, methacholine, or carbachol is usually 1 milligram per mil. Now we are aware that 1 milligram is equivalent to 1,000 microgram. You may want to look up the relationship between milligram and microgram and take note of those things. 1 milligram is equivalent to 1,000 microgram. So what it simply means that this 1 milligram per mil all put together is actually 1,000 microgram per mil. That's what it simply means. Now in the manual for the neostigmine/physostigmine, that's the anticholinesterases, we have 0.1 milligram per mil. But in the laboratory, you may most likely not have this particular concentration. So for you to be able to understand how to work with other concentrations, so let's just use the concentration of 250 microgram per mil. Let us use 250 microgram per mil. So I'm going to be teaching you how to do your calculations in this particular experiment while employing the basics that you have already understood from your first semester pharmacology. Now, one of the things you already know from your first semester pharmacology is that when you are working with the volume of the drugs, your volume should lie within the range of 0.05 to 0.5 mil. This is the range of the volume you must work with. It should not be less than 0.05 mil and it should not be greater than 0.5 mil. So this particular information is going to guide you when you are working with the different dilutions of your solution. So usually in the laboratory, you choose what folds of dilutions you're going to be doing. Usually what we do is 10-fold serial dilution twice. So but then in your alternative to practical, you may be given the number of folds of serial dilution you should do and how many times it's supposed to be done. For example, you can be told to do a 2-fold serial dilution twice, or you may be told to do a 10-fold serial dilution twice, or you may be told that the person who carried out the experiment did a 5-fold serial dilution and then did a 10-fold serial dilution. So you need to understand how all of these dilution terms relate, and it will help you to understand exactly what you are doing. So when we talk about the folds of dilution, what they're actually referring to is the dilution factors that are involved in that particular dilution. So let us look at the formula for dilution factor to give you an idea of what we are doing here. So dilution factor for any form of dilution is usually equal to the final volume over the initial volume. Is the final volume over the initial volume. That is going to give you your dilution factor. The final volume over the initial volume. And another way to calculate for your dilution factor is the initial concentration over the final concentration. So that is for your dilution factor. And then if you are asked to tell what is the dilution you did, your dilution is actually the inverse of dilution factor. That's dilution is equal to 1 over dilution factor. That means dilution is simply initial volume over final volume, and then it's equal to final concentration over initial concentration. So for example, if you have been told that you should do a 10-fold serial dilution on 1000 microgram per mil, it simply means that your dilution factor in that case is 10. And you may want to now calculate, okay, you know that the initial concentration is 1000, but what then is going to be the final concentration since you are doing a 10-fold serial dilution? So what you simply do is to fit it into this formula. Dilution factor for 10-fold serial dilution is 10, and we know that our initial concentration is 1000, and then what we don't know is the final concentration after doing the 10-fold serial dilution. So final concentration cannot be made a subject of formula, so much so that we now have final concentration is equal to 1000 over 10, and that's going to give you 100 microgram per mil. What does this inform you? It tells you that when you do a 10-fold serial dilution on 1000 microgram per mil, you are going to be getting your the next concentration as 100 microgram per mil. Now you know the new concentrations, you cannot be able to tell the volumes you can work with. If it is a 2-fold serial dilution, what you simply do is to put 2 here, and when you put 2 here, 1000 divided by 2 will give you 500. For a 2-fold serial dilution of 1000 microgram per mil, you have 500 microgram per mil as the new concentration, but for a 10-fold serial dilution, you have 100 microgram per mil for the new concentration, just using this particular formula. Then how about your volumes? What exactly are you supposed to do about your volumes? What volumes do you use for you to be able to carry out this particular experiment? Now you can now use the dilution formula to get the volumes you should use. In dilution formula, what we have there is C1 V1 equal to C2 V2. Now what is C1? C1 is the initial concentration. Our V1 is the initial volume, and then our C2 is the final concentration, and then our V2 is the final volume. Our V2 is the final volume. So the initial concentration from what we are given for this particular concentration is 1000 microgram per mil, and then from your calculation you know that the final concentration is going to be 100 microgram per mil. Now what is now left is the initial volume and the final volume. Now, in your normal practicals, you are usually allowed to select what your initial volume is going to be and what your final volume is going to be. But then, in the alternative to practical, you may be told what exactly the initial volume is or the final volume is. But take note, when you are working with alternative to practical, you must know what the question is asking you. If the question will want you to provide all of these things, then you provide it. But if the question does not ask you to go through this particular format, please do not waste your time doing it, because it's not going to get you any mark. For every exam that is being written, there's what is called marking scheme. And if the marking scheme does not capture this, based on the way the structure of the question is being put in place, it simply means that if you waste your time going through all of these, then you are wasting your time getting no mark, because in the marking scheme, it is not captured there. So you must pay attention to your question and know exactly what your question is. So, but basically, we are covering everything, because we don't know what your exam is going to look like. So you have the general knowledge, then in your exam, you now apply specifics as the case may be. So the initial volume or the final volume is usually left to you to decide in your normal practicals. And usually, students will want to use initial volume as 1 ml. Some students may want to use initial volume as 0.5 ml, but it is left for you to choose. 1 ml usually you will have more volumes to be able to work with, but then with 0.5 you have less volume. So depending on what you want to carry out in your practical, it can inform you on what volume to use. So, but for simplicity, we want to use 1 ml as our initial volume. So we're going to be using our initial volume as 1 ml. So the only variable left to be decided is the final volume, which is V2. So how can we calculate for V2? By simply making V2 subject to formula. So V2 is equal to C1V1 over C2. And we can calculate for that now. C1 is 1000, our V1 is 1 ml, and our C2 is 100. This is going to cancel this, and then what we have here is 10 ml. What we have here is 10 ml. So it simply means that the final volume after dilution is going to be 10 ml. So what is initial volume? What is the meaning of initial volume being 1 ml? It simply means we have 1 ml of the 1000 microgram per ml concentration of these particular drugs we are talking about, depending on what you're talking about in the context. So, but then the final volume being 10 ml simply means that you have 1 ml of stock solution. You have 1 ml of stock solution added to 9 ml of the physiological salt solution in that context. But since we are using guinea pig ileum, we're actually working with PSS, which is thyroid. I've talked about that before, thyroid. And in your examinations, please take note, do not spell thyroid for thyroid. So these are two different things. This is an organ in your body, but this is the solution we are talking about here, which is the physiological salt solution. Now we know our final volume is 10, our initial volume is 1. Can we also check the dilution factor to see that it's matching? We know that dilution factor, another formula for it is final volume over initial volume. So 10 divided by 1 will still give you 10. It simply tells you that it's actually a 10-fold serial dilution. It's actually a 10-fold serial dilution. So that's how you work with your serial dilutions. And then for the neostigmine stroke physostigmine, it also applies to it. Depending on the number of folds you intend to get, you know what exactly to apply. Another thing again you want to check is you will be given a particular concentration, but then you need a particular concentration for you to carry out a specific amount, to get a specific amount of the drug that is necessary. Now, I have to distinguish between concentration and amount of drug. When we talk about concentration, we are talking about amounts in volume. So when we talk about concentration, we're talking about amounts in volume. So we have something like microgram per ml, that's concentration. We have something like milligram per ml, that's concentration. So but when we talk about amount itself, we are talking about the amount of drug you are using. It could either be something microgram or something milligram. So you can have 20 microgram of the drug, simply means that is the amount of the drug you are using. So but 20 microgram per ml tells you that the concentration of that drug is 20 microgram in every 1 ml of that particular solution. So it's very important to understand the distinction between concentration and amount of drug. Concentration has to do with the amount in a particular volume, while the amount itself is just the amount without having to attach anything that has to do with volume in it. So for this particular drug, 100 microgram per ml, if you withdraw 2 ml out of this and you're asked what is the amount of drug in 2 ml, it's very simple for you to do the calculation. You already know that 100 microgram per ml simply means that 100 microgram is found in 1 ml. So the next question you ask yourself is how much will now be found in 2 ml. So your S can be made a subject of formula. What you simply do is to do a cross multiply and divide. This times this divided by 1. So what you have is 100 times 2 divided by 1. That's going to be 200 microgram. It simply means that in 2 ml of this particular solution, you are going to be having an amount of drug which is equivalent to 200 micrograms. So it's very important you pay attention to these things while you are working in your practical. So let us just run down through this particular experiment, because the other details, obviously, you have covered them before this time in your first semester. So, but if you have any specific question, do well to ask your question in the comment section, and as soon as possible, we will provide the necessary answers via our media team. So let us make progress in this particular experiment. So let's make progress. Now, you already know how to carry out the experiment, the tracing and all of that. It is very important to take note of that, very carefully. So, but then if you need more help in that, we could always guide you. So, the second thing to do here is to apply, add 10 microgram of acetylcholine. 10 microgram of acetylcholine. If you are going to be getting 10 microgram from 1000 microgram per ml, let us do the calculation. 1000 microgram per ml simply means that 1000 microgram is present in 1 ml, and you need 10 microgram. So how many ml is going to contain 10 microgram? That X is now equal to 10 times 1 divided by 1000. That is going to be 0.01. Yeah, that's going to be 0.01 ml. 0.01 ml is already less than our 0.05 minimum mark. So the implication is that we cannot work with 1000 microgram per ml solution for us to be able to get that amount of acetylcholine. So what we simply need to do is to dilute this guy by any fold we choose, or what is given to us as the alternative in the alternative to practical. But in this particular context, we have done a 10-fold serial dilution where we have 100 microgram per ml. So 100 microgram per ml, 100 microgram per ml simply means that 100 microgram is present in 1 ml. So our 10 microgram is going to be present in X. Our X is now 10 times 1, that's 10 times 1 divided by 100, and that's going to be 0.1 ml. So the implication is that we can be able to get the 10 microgram from the 100 microgram per ml by simply withdrawing 0.1 ml from that particular dilution. So when you withdraw 0.1 ml from that dilution, you apply it to your tissue, and then you get your response. Now the next direction procedure says you should also obtain results for 10 microgram of carbachol and methacholine. Remember I told you that acetylcholine is usually hydrolyzed quickly by cholinesterase enzymes, same with methacholine and then carbachol. Now in this particular experiment, you are going to be reporting the mechanism of action that is responsible for whatever you see as your result. Now when you apply acetylcholine to your guinea pig ileum, it's actually going to act on the nicotinic and the muscarinic receptors, so that by implication you are going to be getting your responses because of acetylcholine being an agonist. That's a parasympathomimetic drug. Same thing with the methacholine and same thing with the carbachol. All of them are going to be acting on both the nicotinic and the muscarinic receptor, leading to the kind of results you now have on your chymograph. So the results you get in your chymograph, it's based on what you've applied. So then the next one we are going to be working with is the anticholinesterase agents, or the anticholinesterases. So what we have here is neostigmine, physostigmine. I told you we have other examples. We have rivastigmine, we have pyridostigmine, and so on and so forth. So, but the concentration of physostigmine or neostigmine that you are giving, will most likely you can have 250 microgram per ml. You can have any other concentration. So, but for the purpose of this particular tutorial video, it is very important that we pay attention to this concentration that is given to us. The next instruction says you should add 10 microgram of any of these agents. So if you're going to be adding 10 microgram, first of all, you have to know if the 10 microgram you're going to be getting as the amount is going to be within the range of the volume you need to withdraw from the stock solution. So we know that 250 microgram per ml simply means that 250 microgram is present in 1 ml. So the next question is 10 microgram will now be present in X ml. So our X is equal to 10 times 1 divided by 250, and that is going to give you 0.04 ml. Now 0.04 ml, based on our range of value for volume, is actually out of place. So the implication is that we have to dilute this further so we can want to get a volume that is within this particular range. You can choose how many fold dilutions you want to do, but for the sake of simplicity, we are going to work with a 10-fold serial dilution, or better still, we could do a serial dilution that is going to reduce it from 250 to 100 microgram per ml. We'll do a serial dilution that moves it from 250 to 100 microgram per ml. So for us to know what dilution factor that is, we want to do something like this. Dilution factor, the initial concentration we'll be having is 250 microgram per ml, and the final concentration we want to get is 100 microgram per ml. So when you divide that, you're going to have 2.5. So it simply means that what we are going to be doing is 2.5-fold serial dilution. Automatically, we're going to reduce 250 microgram per ml to 100 microgram per ml. So what volumes are we going to be using? We'll use our normal C1, V1, C2, V2. So we're going to be using our V1 as 1 ml, so we can calculate what our V2 is supposed to be. So our V2 is now equal to C1 in this context is the initial concentration, 250. We are still using 1 ml as our V1 over our C2, which is 100. So our final volume, based on this particular calculation, so let me just run the calculation quickly. 250 divided by 100. Yeah, our final volume we're going to be having here is going to be 2.5 ml. Now, 2.5 ml is very small for this particular experiment. So we want to increase our initial volume to, let's say, 5 ml. Let us increase our initial volume to, let's say, 5 ml. So let us increase our initial volume to 5 ml, and let's see what it's going to give us in this particular context. So we're going to be increasing it to 5 ml. Remember, I said you're at liberty to choose exactly what to do so that you can get the volume that will be enough for the experiment. So if we change that to 5 ml, we're going to be having 250 times 5 divided by 100. And that is going to give us a very nice volume, which is 12.5 ml. So it simply means that you get 5 ml of the stock solution, and then we add 7.5 ml of PSS, which is thyroid in this particular context, and we're able to get 12.5 ml of the new concentration, which is what? 100 microgram per ml. Now we already have 100 microgram per ml. For us to get the 10 microgram, you simply do 100 microgram to 1 ml. Then 10 microgram will be present in what? And then that is going to give you 0.1 ml. So when you dilute this by 2.5 fold, you get 100 microgram per ml. And the moment you get 100 microgram per ml, the moment you withdraw 0.1 ml from that particular solution, automatically what you are going to be having is your 10 microgram per ml neostigmine or physostigmine, and then you add it to the tissue. Then after adding it, you allow it to act for 5 minutes. After acting for 5 minutes, then you now put in either your acetylcholine first, then the next round methacholine, then the next round carbachol, and also take note of the responses. Now your responses are supposed to be increased for acetylcholine. The reason is because the tissue has some elements of cholinesterase enzymes. So when you added the anticholinesterases, these particular agents, they actually work on the cholinesterase enzymes and decrease the rate of hydrolysis of acetylcholine that is applied to the tissue. Hence, it potentiates the action of acetylcholine. Same thing for methylcholine, because I said methylcholine is actually degraded, but less rapidly than acetylcholine. So, but nevertheless, the neostigmine is going to potentiate its action because it will reduce the rate at which it is being hydrolyzed, and by implication, it will allow for more amount of the parasympathomimetic drug to act very appropriately. So, but for the carbachol, neostigmine has no effect because in the first place, carbachol will not be degraded by cholinesterase enzyme. So, it's important you take note of the mechanism of actions because you're going to be writing that in the inference part of your reporting. So it's very important you take note of that. If you want to look up some of your pharmacology textbooks, Goodman and Gilman, Rang and Dales, and basic clinical pharmacology, and so on and so forth, for you to get more details. So let us now make progress to the next part of the practical, which is the most interesting part. Now, this part talk about having 3 test tubes. And in the 3 test tubes, you have 20 microgram of acetylcholine in 0.4 ml. And then in the first test tube, you are adding 3 drops of blood plus 0.1 ml saline. Second test tube, you are adding 3 drops of blood and then 10 microgram of phystosigmine. And then third test tube, you are adding 3 drops of saline and then 0.1 ml saline. And then you are incubating 3 of them at 37 degrees Celsius for 30 minutes, and then you're administering those contents separately to the tissue. So let me just draw a very quick sketch of these particular test tubes and then writing the components by each side, so I can able to capture it visually, what exactly is contained in the test tube, and I will explain to you what the different components of the test tube will do to the drug that is administered on the particular tissue. So let's make progress. So yeah, what we have here now is the pictorial sketch that helps you understand the components of the 3 test tubes, which I said is very important for you to understand for this particular practical. So let us make reference to the first test tube, test tube 1. Test tube 1 contains, they were told to add 20 microgram of ACH in 0.4 ml. Now let us pay attention to that. It simply means that they have told you the amount of ACH and the volume that should contain such amounts. So what we're talking about here is that we have 20 microgram of ACH present in 0.4 ml. So the next question you want to ask yourself is, if you are having a solution that has 20 microgram of ACH in 0.4 ml, what then is the concentration of that particular solution? So what you simply need to do is to now ask yourself, what amount is now present in 1 ml? Because whenever we talk about concentration in microgram, we talk about microgram per ml. That is microgram in 1 ml. So what amount is present in 1 ml? So can we guess for X? X is equal to our 20, remember our cross multiplying, 20 times 1 divided by 0.4. If you do that in your calculator, what it's going to give you is going to be 50 microgram. So it simply means that the concentration of the drug that you were able to get 20 microgram in 0.4 ml from is a solution, ACH solution with concentration of 50 microgram per ml. Now remember that we have initially reduced from 100 microgram per ml, 1000 microgram per ml to 100 microgram per ml. But then we are now moving from that 100 microgram per ml to 50 microgram per ml. In case you're asked, okay, how many fold dilation is that? It's very simple. Final volume over initial volume of initial concentration over final concentration. So that is 100 initial concentration divided by 50. That's going to be a 2-fold serial dilution. So we do a 2-fold serial dilution, gets to 50. Then we withdraw 0.4 ml, which contains 20 microgram of acetylcholine. Remember what I said. We have reduced this from 1000 to, let me write that. We have reduced this from 1000 to 100, and that is a 10-fold serial dilution. Then from the 100, we now reduce it to 50 because we need to withdraw 20 microgram, which is present in 0.4, and it's only 50 microgram per ml concentration that we're able to give you such amount. So we take it to 50, and that is a 2-fold, and that's a 2-fold serial dilution. That's a 2-fold serial dilution. So it's very important you understand how these things operate, because your alternative to practical questions may want to play on these values, and you will need to be as smart as possible to do very well without having to break down in the examination hall. So what we have here is 20 microgram of ACH in 0.4 ml of the solution, then plus 3 drops of blood. The question you want to ask yourself is, what is blood doing here? Yeah, blood is doing a very, very simple job. Blood contains cholinesterase enzymes. So in the blood, you have cholinesterase enzymes. So addition of blood is simply as good as adding cholinesterase enzymes. And that's exactly why you're having blood in this particular contest. And then you have your 0.1 ml of normal saline. Normal saline, I told you, has no effect. It's just neutral. It's isotonic with the blood. So it's not going to do anything to the particular reaction. Now, we know that cholinesterase enzymes rapidly rapidly hydrolyzes acetylcholine, causes its degradation. The implication is that the blood added to this ACH is going to decrease the concentration, the contraction, that's the response you're going to be getting from your ACH. If ACH normally should give you a contraction of, say, 5 millimeter, and then you are now adding ACH with blood that contains cholinesterase, then 5 millimeter is no longer going to be your contraction. You're going to be having a lesser contraction because you now have a lesser amount of ACH available to act on the tissue by reason of degradation by the enzyme, the cholinesterase enzyme. And also remember that your procedure tells you that you should incubate at 37 degrees Celsius. The reason is because your body temperature is around 37 degrees Celsius, and by incubating at 37 degrees Celsius, you allow the enzyme to act at its normal temperature. So by implication, you allow that cholinesterase enzyme to act on the ACH because it functions best as the temperature of the body, which is somewhere around 37 degrees Celsius. So the implication is that for this particular test tube, when you apply it to the tissue, you are going to be having a decreased contraction because the blood containing cholinesterase enzyme is going to cause hydrolysis of ACH. But if what you have here is methacholine, the contraction will be decreased, but not so much as you have in ACH. And if what you have here is carbachol, there will be no decrease in contraction because in the presence of cholinesterase, carbachol is immune. It is not destroyed, it's not dehydrolyzed, it's not degraded by cholinesterase present in the blood. So all of these things are based on the theories that we have concerning this. And when you are carrying out the practical, it becomes more fun. So let us look at test tube 2. Test tube 2 is still 20 microgram of ACH from 0.4 ml. Remember, we are using a solution with concentration of 50 microgram per ml, two-fold serial dilution. I already explained that earlier. And then you are adding that to 3 drops of blood, and you are adding 10 microgram of physostigmine in 0.1 ml. If you go back to the calculation we did for physostigmine 10 microgram, when we moved this 250 to 10 microgram to 100 microgram, you can already see that from the 100 microgram per ml, you can get 10 microgram of physostigmine using 0.1 ml. You may just want to go back in the video a little and you see the calculation when I explain this at this particular part. It will help you to understand that the 10 microgram of physostigmine, we don't need to dilute again because we already have a 100 microgram per ml solution of physostigmine that we can withdraw 0.1 ml from, that contains the amount of 0.010 microgram of physostigmine. Now, what is physostigmine? An anticholinesterase. So it simply means that physostigmine will react with the cholinesterase enzymes in the blood while you are incubating for the 30 minutes, and then by implication, it means that the contraction you will have from this test tube is going to be higher than the contraction you have in this test tube. Remember, some of the enzymes will still act on the ACH. Yeah, and then it causes breakdown of some of the ACH. But the breakdown you have here will not be as much as the one you have here because you have an anticholinesterase that is preventing the action of the cholinesterase enzyme on the ACH. And like I told you, carbachol is not going to be affected by this. And then when you come over to this, we have 20 microgram of ACH, 3 drops of normal saline, 0.1 ml normal saline. Normal saline does nothing here. So this test tube 3 is going to be serving as our control for the experiment. So I can be able to monitor the things that influence the results you get from this particular experiment. So when you have mixed all of these things together, you incubate them for 30 minutes. I already explained why you need to incubate it, because the enzymes function at the temperature of your body, which is around 37 degrees Celsius. And then you pour it into the tissue. You are going to be getting responses according to the theories I just explained for you. So now let's rush or go quickly into the report of this particular experiment. So when you are reporting this experiment, you have the drugs and doses administered. Is in your manual, so you can see that their tissue response. Then under it, you have the height. The height can be in millimeter, the height can be in centimeter. So you must inquire from your coordinator to know which unit you must use. If you use millimeter or centimeter. During my time, we use millimeter, but then your coordinator tells you what to do because that's what him or her is going to be working with in your marking scheme. Then we have the inference. The inference is where you explain the mechanism of action behind what you just carried out. And that is why you must understand the basis of autonomic pharmacology. That's why you must understand how these drugs work on the sympathetic and the parasympathetic nervous system. So let us start. Let's just run 3 different reports of drugs or doses administered. Let me show you how to report your experiment. But your coordinator will give you more emphasis. If him or her has any unique attributes, you will have to add to your table. So 10 microgram of ACH plus tissue. Did you see that 10 microgram of ACH is added to tissue? So the result of it is going to be contraction. And then the height of response is 5 millimeter. What is the inference? The tissue contains muscarinic and nicotinic receptors. ACH will bind to the muscarinic and nicotinic receptors to give the desired effect, which is contraction. You want to take this inference a bit longer, but in the case of alternative to practical, this is already sufficient for you because you don't have all the time in the world to report your experiment. So you just have to be as precise as possible and also ensure that you are straight to the point. But doing normal reports, we want to be as extensive as possible so I can get as much marks as is allocated for that particular explanation. Then the next one we have is 10 microgram of physostigmine anticholinesterase acting on the tissue, first of all, and then you're now adding 10 microgram of ACH. Now, what happens? Increased contraction, because the contraction have increased from 5 millimeter to 9 millimeter. So you don't just write contraction, you write increased contraction. Why is there increased contraction? That's why you're going to be writing in inference. It says physostigmine is an anticholinesterase and it will bind to cholinesterase enzyme that's already present in the tissue, the residual amount of cholinesterase enzyme in the tissue, and it will allow for more ACH to act on the tissue. And the end result is that you're going to be having increased contraction. Then let us come to this particular one. We have 20 microgram of ACH, 3 drops of blood, and 0.1 ml normal saline incubate plus tissue. You can see every single step we took is captured under the drugs and doses administered. So anybody that is reading your report of your experiment will know exactly what you did and what led to what. Now, the result of this particular one is decreased contraction, and then we're having 2 millimeter. The next question is, what is your inference? I already explained that earlier on. The blood contains cholinesterase enzyme, and by acting on ACH, it degraded the ACH. So by implication, the contraction you're going to be getting for ACH is going to be less than the contraction you have when you have ACH acting on the tissue alone, or when you have ACH acting on the tissue together with an anticholinesterase agent. So that is that about this particular experiment. So in the next videos, we're going to be talking about parasympathomimetic drugs, that is the antagonist, and we're going to be looking at other unique areas of this particular pharmacology practical for you in the second semester. So stay tuned and subscribe to this particular video. Thank you so much.